Morning, everyone. For our Physics 31A presentation, we will be discussing the physics of flowering plants and pollinators.
Hello everyone, my name is Kyra and my planned future career is to become a doctor.
My name is Nicole, and my passion lies in ecology and sustainability, and I plan to major in environmental science.
You might be wondering how flowers are related to the medical field, and the simple answer to that is through plants. Certain parts of plants are used in the medical field to aid in treatment or creating medicine. An example of a plant is the opium poppy. The white crystal extracted from opium poppies can be used as morphine, which is a strong painkiller used after medical procedures. There are two specific plant extractions that have a physics component: microwave assisted extraction and ultrasound assisted extraction. Microwave assisted extraction is normally used for flavonoids. Typically, electromagnetic radiation is emitted with a frequency of 300 megahertz to 300 gigahertz and a wavelength between 1 centimeter to 1 meter. Once microwave radiation hits an object, the object absorbs this energy and converts it to heat, which helps bring the solvent to the drug matrix so the extraction process can be complete. Ultrasound assisted extraction uses sound energy at a frequency higher than 20 kilohertz, so it will interrupt the plant cells and increase the drug surface area for solvent extraction. The highest sound energy quickens the extraction process because it minimizes the heat needed for the extraction to occur.
As you might have guessed, studying flowers and pollinators is extremely important when it comes to the field of ecology. Their purpose in facilitating pollination and seed dispersal is crucial when it comes to increasing biodiversity both in natural and urban areas. Flowers and accompanying pollinators also contribute to helping 75% of our crops produce the fruits we consume. So some background on angiosperms, otherwise known as flowering plants, first arose around 125 million years ago. With 300,000 different species that make up 80% of all our plants, our presentation seeks to learn how flowering plants utilize the laws of physics to shape the course of their evolution and how they might influence the development of our technology. First and foremost, why are flowers so colorful? Flowers produce two major classes of pigments to help attract pollinators, carotenoids and flavonoids. The set of wavelengths absorbed by a pigment is its absorption spectrum, and the reflective light is the color we see. Carotenoids, which are responsible for reflecting yellow, orange, and red colors, absorb violet and blue-green wavelengths at around 400 to 500 nanometers. Meanwhile, flavonoids, which include anthocyanin pigments, create red, purple, magenta, and blue hues and absorb blue-green light at around 450 to 580 nanometers. The color we see in flowers is characterized by the frequency of photons, which means how many wavelengths a photon propagates each second. The higher the frequency, the more energetic the photon. On the other hand, longer wavelengths are associated with less energetic photons. Flowers also rely on structural colors to give off a form of iridescence to further attract pollinators. Structural colors rely on microscopically textured surfaces that are fine enough to cause specific wavelengths to interfere constructively or destructively. Researchers have found that the mixta gene is responsible for forming a conical geometry in the epidermal cells of flowers where the areas are directly exposed to pollinators. This shape acts as a lens by focusing light into vacuoles containing anthocyanin pigment. In turn, it allows a flower's color to become more intense without having to produce more pigment. A sparkling appearance also scatters light from the inner tissues much more efficiently. Conical cells also increase frictional force between the pollinator's tarsal claws and the plant surface. On the molecular level, the bumpy surface of a petal can interlock with the hairs of an insect's leg, making it harder to move them past each other. The more bumpy and textured the surface is of the flower, the greater the frictional coefficient it has, and thus a greater frictional force. Similarly, some flowers lose the cone cells for a flatter surface when birds are the main pollinators. A flatter surface minimizes friction and therefore nectar-robbing insects because it causes them to slip more easily.
Now we will talk about the variety of pollination processes and the physics behind each mechanism. The first mechanism is with bunchberries. I will play this short video clip so you can visually see what it looks like. Bunchberries produce a lot of force for this catapult to occur. These bunchberries have petals that are enclosed around the stalked anthers that contain pollen. As the plant grows, the filaments, which are the anther stalks, lengthen faster than the petals, making the filaments bend and store elastic energy. When the petals open, this stored elastic energy is released, and the pollen goes from rest to having a high velocity. This catapult can land on the pollinator in a projectile motion, and the pollen can also be blown away. Projectile motion would be that pollen's speed increases as it goes up, or the projectile motion would be that the pollen's speed decreases as it goes up and increases as the pollen grains go downwards onto the pollinator. During this time, gravity acts as upon the pollen. The next pollination type is buzz pollination. The flowers need to be shaken because the flowers protect its pollen from other insects by keeping the pollen in the anthers, and the pollen does not fall out easily. When the bee violently shakes the flower, the pollen is released due to floral vibrations caused by the bee shaking the pollen using their thoracic muscle, which spreads throughout the bee's body, creating a buzzing sound. This buzz can be one continuous vibration or multiple short vibrations, but the vibrations will be uninterrupted. The floral vibrations have a fundamental frequency of around 100 to 400 hertz. These vibrations felt by the anthers depend on the stiffness, mass, and material of the flower. The larger the stiffness or mass, the harder it is for the anthers to feel the vibration and release pollen. There has also been a bit of research done to state that the kinetic energy buildup from the internal walls of the anther and the pollen grain's elastic collisions assisted in the release of pollen as well. Stylidium plants also have a unique pollination mechanism. The stamen and style are fused together to form a column which stands upward. It is able to stand upwards through changes in the volume and length of cell, which occurs from the potassium ion transport. When the insect brushes the flower, the stored energy from this column releases, and the column snaps downwards, so the pollen lands on the pollinator. When the column is still, its stored potential energy is at its highest level. However, once this column moves to deposit the pollen on to the insect, the potential energy slowly shifts to kinetic energy until the column goes back to rest, where the kinetic energy shifts back to potential energy. Now I will talk about electrostatic forces involved in flower pollination. First, charged pollen is able to settle onto the stigma of the flower better than uncharged pollen, which is important for fertilization. Electrostatic charges accumulate on flying pollinators, and these charges start to create an electrical field between the flower and the insect. The flower also has a charge, so the electrical field is stronger when the distance between the flower and insect decreases. This electrical field also aids in the transferring of the pollen from the anther to the pollinator and the pollen from the pollinator to the stigma. Hummingbirds have enough electrical charge to track pollen grains and bend stamens. Bumblebees can detect the electrical field between themselves and a flower through their mechanosensory hairs. The flower's electrical field will depend on its geometry of the flower, which is tested by spraying electrically charged paint on the flower so the electrical field of the flower is visible. Lastly, the electrical field of the flower changes before a bee lands on it, and this change lasts for a few minutes. On the right side is a picture of the parts of the flower, such as the stigma, stamen, anther, and filament. On the left side is a picture of the charges surrounding the pollinator and the flower. The flower has a negative charge, and the pollinator has a positive charge, and because the charges are opposite, they attract each other.
Moving on to bats, Macravia, Avenia, and echolocation. M. avenia is a tropical vine endemic to Cuba that uses concave leaves to amplify acoustics to attract pollinating bats. When bats emit an echolocation sound, they can distinguish that echoes bounce back to them because the spherical gap geometry, aka its concavity, allows the plant to return a strong and consistent echo over a wide angular range. The leaf center and edge reflect sound differently, but in a way that overlaps and reduces distracting background noise. In addition to its satellite dish-shaped leaves, their flower/nectary is also very waxy and thick, which also aids in amplifying sound waves back to the bat. Researchers from Antwerp University have also taken inspiration from the echo vine by scanning and 3D printing their own reflectors. A robot using this bio-inspired reflector was able to steer its way past unknown terrain solely by sonar and using the reflectors as waypoints as seen in this video to the right. The aerial view shows the different size reflectors spread throughout the room and how the robot was able to pinpoint their location based on its reflected sound. Yay! Thank you for
Yay,
listening.
thank you for listening.
Happy finals. And citation.
Citation.